Hello there and welcome. This is Lecture 1 for Anatomy and Physiology 2, a hybrid course at River Valley Community College. And we'll be beginning Unit 1, which is about the cardiovascular system. And today's lecture is about blood and the composition of blood. So the first thing we need to know is what the function of blood in the cardiovascular system is. Basically, blood is a connective tissue, and it doesn't connect things in the same way as a ligament or tendon would, but it does connect distal parts of the body to one another. And it does that by carrying oxygen, wastes, including carbon dioxide, nutrients to and from the tissues and the cells. So it basically takes good stuff and gets it where it needs to be, and takes bad stuff away from the cells, basically metabolic byproducts, things like uric acid, ammonia, hydrogen ions, things like that, carbon dioxide, away from the tissues and the cells, and allows them to be eliminated either by the kidneys or through the lungs or whatever the case may be, depending on the various types of waste we talk about. Another thing that blood does is it carries hormones to target organs. So if we think of having an adrenergic response, so when we talked about the sympathetic nervous system, when adrenaline gets going, the effects of that adrenaline are very widespread. Your heart goes faster, your breathing changes, your tone of your digestive tract and your muscles, blood vessels in your skin, all of those different things are changing, but they're all changing in response to a single hormone. And so that implies that the blood is carrying to all of those different and disparate parts of the body that hormone is getting there. And so that's one of the functions that we think about when we think about blood. And then last, blood's really important in the maintenance of homeostasis. So it's important in temperature regulation. As I mentioned, vasoconstriction, vasodilation are ways that we allow our bodies to either lose or retain heat. So if you're hot, you flush in your skin because blood is going to the superficial parts of your body, just like a radiator in a car would. And if you're cold, then you vasoconstrict. So we keep that warm blood away from our skin in order to be able to help regulate temperature. It's also just got a big thermal mass when we think of the amount of blood that's in the human body and the proportion of that blood that's water. Water has a very high specific heat, meaning it's slow to change its temperature. And so having so much kind of watery substance in the body is one of the ways of just kind of having a temperature inertia, if you will. It keeps it from changing temperature very readily. Blood is also controlled within a very narrow pH range and allows the body to regulate pH in times of exercise. For example, when we have carbon dioxide building up in the blood, we get rid of that by off-gassing, increasing our respiratory rate. So there are a lot of different things that are going to be important in terms of pH. Osmolarity, we'll see a very frightening example of in just a moment, but osmolarity is basically the fluid balance of the body. Keeping the liquid portion of the blood where it needs to be is an important job of the blood. And then hemostasis, which is the ability to stop bleeding. And so that's one of the things we'll talk about in some detail about how platelets work in order to stop bleeding and maintain homeostasis in the blood. And then lastly, to prevent infection. And so that's really the main job of white blood cells in their various forms, is to fight off pathogenic organisms that may cause infection in the body. So as I mentioned, osmolarity is one of the really important things that happens in the blood, and this is an example of a homeostatic imbalance. In other words, when things aren't going right in the blood, what can happen. So this is called ascites, and as you can see, this is a man on the right-hand side, not a pregnant woman, and the ascites that you see, and you can see in this image here, and this is not the same belly. This guy's belly and this person's belly are not the same, but gets the point across that when the liver is not making proteins, specific proteins properly, those proteins are lacking from the blood, and the osmotic gradient in the blood isn't sufficient to keep the liquid part of the blood in the blood, and so it leaks out into these kind of interstitial spaces. So this is what we would call the peritoneal space So the peritoneum has a visceral and parietal layer like most membranes in the body, right? So you have the visceral layer that's on the outside of the organs, that's here, and then the parietal layer that's here. And so the fluid part here is leaking out into that space, and that's all caused by a problem in the composition of the blood that is usually associated with liver failure, and most often that liver failure is associated with cirrhosis of the liver as associated with alcoholism. So what's in the blood and how does it all work? So first of all, the blood is basically, I think of kind of like a snow globe or one of those lazy rivers that you would have at like an amusement park, that it's got this non-living liquid, which is basically extracellular matrix, like all connective tissue. It's got a lot of extracellular matrix, and that's called plasma. And that is basically the liquid component of the blood. And then floating in that snow globe or floating in that lazy river, you have some living cells, and those living cells make about 45% collectively, with the vast majority of the formed elements, the living cells, being erythrocytes or red blood cells. And then the white blood cells or leukocytes and platelets collectively make about 1%. So if you think about it being 45%, this could be basically 44 and 1. But as you'll see in a minute, hematocrit is usually around 45%. So these are all estimated numbers, but should give you a pretty good idea of the composition of blood. So when you spin blood down in a centrifuge, what you get is the cellular components, which are heavier, settling to the bottom, just like if you had that snow globe and you shook it up and you let it settle, then you would get the heavy stuff going to the bottom. And so you'd see that you would have red blood cells at the bottom. Then you have a layer of white blood cells and platelets here just on top, and that's sometimes called the buffy coat. And then above that you have the liquid portion or the plasma of the blood. And you should be familiar with this picture both in terms of what goes where and about how much of it there is. So you can see here we've got about 4.5 milliliters of cellular components at the bottom out of 10, and then we've got about 5.5 milliliters of plasma here, and that makes about that 55%. So when we look at plasma, which is what we're about to do, we see that it's a vast majority of it is water, but then it's got some proteins and some nutrients, hormones, and electrolytes inside of it, and that kind of makes that concoction that's nutritive to the cells. The water really facilitates its movement throughout the body. So if we don't have enough, if we're dehydrated, some of the things that happen are a result of not having enough water in the plasma. And those things are all important. Electrolytes, so things like sodium and potassium are very important, calcium, blood components, and they're all carried in the plasma. And then in the cellular components, which we'll talk about in more detail in a little bit, we have the buffy coat and the red blood cells, and we'll talk about each of those components in a little bit. So the plasma is contained within a very, very narrow pH range, 7.36 to 7.45. If we get above that, it's called alkalosis. And if we get below that, it's called physiologic acidosis. Physiologic meaning that it's not true acidosis because we're still above 7. But it's acidotic relative to what the body is capable of dealing with. So that's got to be pretty tightly regulated. The plasma is slightly warmer than regular body temperature, so that's one kind of interesting thing. And it's got some proteins in it, but by far about 60% of the protein contained in the plasma is albumin, which is the most abundant. It's one of those things that's made by the liver, and that's why when people get ascites, that's because of albumin, among other things, not retaining that osmotic gradient going into the blood. And then another really important protein that you'll learn about in clotting and hemostasis is fibrinogen, and that's something that's kind of circulating in lower concentrations, but is also contained in the plasma. As we know, we have ions, hormones, and nutrients. One of the big nutrients that we're going to talk about is glucose in the blood. So the formed elements are basically the living parts, okay, that are floating in that non-living cellular matrix, the plasma, and they make up about 45% of the whole blood, and come in three basic cell types. And that's a bit of a misnomer because only leukocytes are truly cells, but erythrocytes are what we call red blood cells, and they're anucleate, meaning they have no nucleus. They don't really have anything else going on except being bags of hemoglobin. We'll talk about that in a minute. Leukocytes are the true cells. They're white blood cells. And then platelets are actually fragments of an exploded megakaryocyte, which is a huge cell that breaks up into lots of little pieces, and those pieces collectively are platelets. So one of the things that we're really interested in when we talk about the formed elements is something called the hematocrit, and that's the percentage of a person's blood that is red blood cells. And that's really important clinically because red blood cells, as I mentioned, are the things that carry oxygen, and we want to have a sufficient number of red blood cells in order to be able to adequately oxygenate the tissues. So the normal range for hematocrit, a normal hematocrit is about 45%, as you saw in that first slide. 45% of our blood should be red blood cells. And it tends to be a little bit higher in men, both because of the action of testosterone and because of the lack of menstruation. But men have a hematocrit about 47% plus or minus 5, where females have about 42% plus or minus 5. And again, that's because testosterone kind of amps up blood cell production by activating erythropoietin, and in women, menstruation is a source of blood loss. So it's typical for women to have a little bit lower hematocrits than men. So what's the deal with erythrocytes? So first of all, there are a lot of them. Okay, they outnumber white blood cells by about 1,000 to 1. And they are really well suited, and I think your textbook goes into this in some detail, in terms of their link between structure and function. So how is that? Because we always care about structure and function a lot in this class. And one of the ways is they have a very large surface area to volume ratio. Part of that is because they're kind of flattened discs, but then to even add to that, they're biconcave, so they're kind of indented like a collapsed jelly donut on either side. And that allows them to really have a large surface area relative to their volume. And then basically there's nothing else in a red blood cell except for hemoglobin. And hemoglobin is a really cool molecule in that it has a high affinity for oxygen, but not so high that it won't let go of it once it's bound, because it has to be able, it's kind of like if you took a rescue dog in or something like that, you have to be able to be attached to it and take good care of it and everything like that. But then when it's time to give the dog over to somebody who's handicapped perhaps and needs a service dog, if you've raised that dog, you need to be able to kind of let go of it and give it on to somebody else. And in the same way, hemoglobin takes care of the oxygen, chaperones it to where it needs to be, and is very attached to it during that time, but it doesn't have such a high affinity that it won't let it go once it gets to the tissues. And that's a really important consequence or characteristic, I should say, of hemoglobin. So each red blood cell can carry about 1 billion oxygens. And if you think of how many 4 to 6 million red blood cells we have, it's a heck of a lot of oxygen that we're capable of carrying. The other thing that's interesting is that because the hemoglobin is basically the only component of an erythrocyte, there are no mitochondria, there are no kind of cellular apparatuses that are using oxygen. And so, pardon the saying, but basically if the goods that are being carried are oxygens, there's no utilization of oxygen by the red blood cell itself. And so it's able to really carry it and drop it off without kind of tapping into the supply. So that's that. So here's just a picture of hemoglobin. Basically, you should know that there are 2 alpha chains, okay, and 2 beta chains here, and that the heme molecule is embedded in each of those. These are just basically protein sheets. So if you guys remember alpha helixes and beta pleated sheets, those are kind of the deal here. But these molecules of heme here have as a central atom an iron atom. And that's really important when we talk about anemia, which we're not going to get to in this lecture, but we will very shortly. So we'll start here with anemia in the next lecture, and hopefully that's been a little bit of an introduction to the composition of blood.